Alrighty. Hello everyone, welcome back to PS Platypus. Today we're going to be having a look at the histology of the renal system. So without further ado, let's get started. So we're actually going to be having a look at the histology of the entire urinary tract, starting up top with the kidneys, having a look at five main structures there: the corpuscles, proximal convoluted tubule, distal convoluted tubule, loop of Henle, and the collecting ducts. Then we're going to have a look at the ureters, and finally we're going to end by having a look at the bladder. So all of that will be covered in this slide set. So let's start off with the corpuscles. Now, the corpuscles are sort of the initial part of your nephron, and they consist of your glomerular arteries and Bowman's capsule. Now, when it comes to the corpuscles, the very first thing you should be able to do is figure out what they look like so that you can immediately identify them on histology. And I think one characteristic that you can use is the fact that they look a bit like globes or balls on histology. So you can sort of see them very clearly when you're given a histological slide of a kidney. The next thing you want to be able to do is orient yourself to which side is which. So you can see here is sort of the capillary network right there. And then the Bowman space, which is where all of the fluid from the capillary network will be draining into. And you can see that sort of stylized in the image on the bottom right there. I think that's a better description of that. And then probably the most important part is to be able to identify what's called the urinary pole. So you can see where all of this fluid sort of drains down and into the proximal convoluted tubule. That's called the urinary pole. And opposite that is called the vascular pole, because that's the end where the vascular network, the capillary network, the glomerular capillaries meets the nephron. So keep that in mind. The other thing to keep in mind is that the vascular pole is also the location where, as I mentioned before, the afferent and efferent arterioles are entering and exiting the glomerulus, and also where the distal convoluted tubule actually winds back up to touch the glomerulus again. If that sort of doesn't make sense, have a look at this diagram here. You can see this glomerulus right there, and you can see there's an afferent arteriole entering and an efferent arteriole leaving. And you can also see that if you follow the nephron back around, up, down the loop of Henle, the distal convoluted tubule actually comes back and touches the vascular pole, the site where those arterioles are entering and exiting. And you can't really appreciate that as well, maybe on this slide, but it does exist at the vascular pole. The next thing you have to keep in mind is these things called podocytes. And again, you'll see better images of them in your histology lessons where you have sort of zoomed up versions of them. But all you have to really know about them is that these specialized cells that have foot processes that are important for forming the filtration barrier, which we'll come to in a second. And that actually brings us to this image on the left. So if you were to zoom into the interface between the glomerular capillaries and the Bowman space, you'd see this filtration barrier. And that's the barrier that filtrates must pass through to go from the capillary lumen into the Bowman space. So you start off from the capillary lumen, and then to be able to enter the Bowman space, the very first barrier you'll come across is obviously going to be the lining of the capillary itself, which in this case is a specialized epithelium, which we call endothelium. So the capillary's epithelium, endothelium. In this case, the endothelium is fenestrated, so it has gaps in it that allow certain substances through. And then once it passes through the fenestrated endothelium, the next barrier it encounters is going to be the basement membrane, because we know that under all epithelial surfaces or under all epithelial cells, you also have a basement membrane. That's going to be the next barrier it comes across. And then after that, we have those foot processes from the podocytes forming filtration slits. And that's going to be the final barrier it comes across before it can finally cross and enter the Bowman space and then drain into the proximal convoluted tubule. And you'll cover in more detail about what sort of substances can go through, what can't, in both your physiology lectures and also the lectures you'll have at Monash, where you'll go into more detailed histology and linking that up with different conditions as well. All right, so next up, with regards to the kidneys, we're going to be having a look at something called the juxtaglomerular apparatus, also abbreviated as the JG apparatus. And you can see that over here. Essentially, on histology, they won't ask you this too much, but you can also identify important cell types that form up that JG apparatus. So the macula densa is going to be up here at the urinary pole. And then you also have mesangial cells and juxtaglomerular cells, all of which can be appreciated very hard to see sometimes on histology, but you can appreciate it in the stylized version of the diagram on the bottom right there. And again, you'll cover in more detail the physiology of how the juxtaglomerular feedback and apparatus works in more detail during your physiology lecture. So I won't go through it here, but keep that in mind as well, that you can see it on histology. Next, we come to the tubular network. So the proximal convoluted tubules, the loops of Henle, and the distal convoluted tubule. So just to remind you, what happens is the filtrate that came out of the glomerulus or the glomerular capillaries into the Bowman space, then drains into the proximal convoluted tubule, goes down the proximal convoluted tubule, down the thick and thin descending and ascending limbs of the loop of Henle, and then up the distal convoluted tubule, and then into the collecting ducts. So we're going to be having a look at that tubular network over here. Now, some important things to keep in mind is how you can differentiate between them in histology, and also how they are different in terms of cell type. So in terms of the cell types that form these different tubules, with the proximal convoluted tubule, all you have to know, same with the distal convoluted tubule, is that they're both simple cuboidal. With the loop of Henle, however, it's a bit different. And that makes sense because the thin part of it is actually going to be made of a more squamous or flat, squashed shape epithelial cell rather than cuboidal. So that's all you have to remember, is that pretty much all the tubular networks are simple cuboidal, the only exception being that the thin limbs of the loop of Henle are formed of a squashed squamous cell, simple squamous cells. The other thing to keep in mind, which they can often sort of quiz you about, is how do you tell between the proximal convoluted tubules and distal convoluted tubules? Because just on that fact, you can't really tell them apart. And that comes down to two main things. The first one is that with the proximal convoluted tubule, you'll often see what's called a brush border. And that can be kind of, again, hard to see on histology. But for example, in the image over here, you can see sort of a fuzzy surface on the luminal side of the epithelium. And you can also see it over here with all the proximal convoluted tubules, as well as in this sort of clearer diagram where you can make it out right there. And on the other hand, distal convoluted tubules don't really have that brush border. It might be a little bit fuzzier because of the way the histological image was taken, but you can kind of sort of clearly make out the luminal surface. And so that's one way you can differentiate between the two. The next way you can differentiate, but again, depends on the type of staining they use and like how strong that staining was, is how darkly it stains. So with proximal convoluted tubules, you have sort of a more acidophilic and darker staining of it because more absorption is happening here. And so you can see on this image, for example, that the PCTs are all very dark and intensely staining, whereas the DCTs are sort of lighter. But again, if you look at the real histological images over here, that's sort of harder to appreciate. So it's not always a great way to differentiate the two, but you can use it sometimes. And another thing that you might have as a result is because the PCTs, they stain more darkly and intensely, they often have nuclei that are less discernible because the stain of the cytoplasm is much more strong. And that's another thing you can use. So for example, if you look at the PCTs here, the mitochondria is kind of almost like covered in a film of cloudiness, whereas the DCTs, you can see the mitochondria a lot more clearly. So that's one thing you could use. But if they ask you what the differences are, you can point them out as being brush border presence in PCT, darker stain for PCT, as well as less discernible nuclei with the PCT. All right. Next, we come to the collecting ducts. So from the tubular network, your filtrate goes up the distal convoluted tubule and then finally drains into the collecting duct. And the collecting duct is, again, also simple cuboidal, and that's written in the speaker notes, by the way, if you want to have a look at that. But then from the collecting duct, it'll drain into the renal pelvis, and then from there, enter into the minor calyces, major calyces, and make its way into the ureters. All right, and here's a bit of an overview of everything we've covered so far. So these diagrams sort of try and put everything into one concise little slide. I think what's really useful as an exercise is to be able to look at the image over here and see if you can identify what we've talked about so far. Probably the main thing you want to be able to point out straight away is where the glomeruli are. And thankfully, you can see clearly that all those ball-shaped structures, right, over here, here, they are very clear and easy to see, and they indicate glomeruli. The PCT, LOH, DCT, you can sort of make out, but you'd only be able to differentiate really if you could zoom in a little bit more and see those structures on the inside. And then the collecting tubule or collecting duct, you can make out sort of at the bottom there. Then we come to the ureters. So we've got our filtrate from the kidneys, and now we're putting them into the ureters. And now we come to a change in the epithelial type. So before we had simple cuboidal primarily, a little bit of simple squamous as well. But now we're coming to transitional epithelium. And so this is epithelium that can sort of flatten and give more, increase the volume inside of its lumen so that it can accommodate more urine, for example. And we'll have a look at that in the next slide as well, exactly what that looks like. But the next important thing you have to know about the ureters is that it's surrounded by a layer of smooth muscle. And you can sort of make it out here on the outside, this pinkish histological stain. And that muscular layer allows the ureters to undergo peristalsis, which is important because it allows to propel the urine forward, especially if you have obstacles in the way. It can allow it to squeeze past that obstacle and even clear, small stones and small obstructions as well. I guess the only important thing, the high yield can take away from here is you should be able to see this image and sort of this distinct sort of folded structure and be able to immediately identify it as the ureter. And the good thing is it's quite easy to identify as opposed to some of the other more difficult and harder to differentiate things like the PCT and DCT. This is something you should be really easily able to rattle off as being the ureter. And then we come to the bladder. And this again is lined by transitional epithelium, but in this slide, we sort of show exactly what that means. Transitional epithelium is, as I said before, it's epithelium that's able to collapse and stretch in response to pressure and thus increase the capacity and volume of the lumen that it's surrounding. So for example, when the bladder is empty, the transitional epithelium isn't collapsed, it's sort of staying up like that. It's in its relaxed state. But then let's say the bladder starts filling up and it needs more and more space for the urine to occupy, the epithelium can accommodate by collapsing and creating more space for the urine to occupy. And that's sort of the advantage of having that in both the bladder and the ureter. It's also surrounded by smooth muscle, which you'll learn more about again in anatomy, in physiology, and the way that it's wired up to the autonomic nervous system. But we won't go through that in too much detail here. All right, so that was a lot of content, and I know it can be a little bit overwhelming. I guess the main thing with histology is that it's all about pattern recognition. You should be able to have a look at these images enough time so that when you see them again, say for example, in the exam or in quizzes, you should be able to quickly know what's what. So, if you see those ball-shaped structures, you should know they're glomeruli. If you see that sort of star-shaped lumen, you should see it's like sort of a ureter. And so that's really, really important that you practice. And because it can be hard to find good resources to practice with, I've linked to a couple that I found useful when I was studying histology. So there's 2 really good histology quizzes online that I used. Some of the questions, again, can be a little bit more advanced, so just skip them and don't worry about them too much, but they are good references. The second one there isn't really a histology quiz. It's more of a freestyle thing where you can go onto a histological slide of the kidney, zoom in, scroll through, move around, and see if you can start your way from the outside of the kidney and work your way in, identify some of the key structures there as sort of a fun little exercise to see how much makes sense and how much maybe you need to revise a little bit more about. Then in terms of textbooks, again, textbooks can be a bit overwhelming because they contain content that might not always be useful or might not be stuff you've covered at all. So in terms of options, one that's recommended is Lippincott's review questions. And again, you can find a copy of that on the Moomoo's drive. Again, over here, focus only on the questions you can attempt and disregard some of the more difficult higher end questions that are way beyond the syllabus that you're going to be covering at Monash. And then, of course, the gold standard for what you should expect in the exam is going to be past exam papers. So have a go at them. They're a really reliable and accurate representation of what you can expect in your own exams. And in preparation for your mid-years, your end of years, you should take a look at them so you can really understand what sort of stuff they can ask about in histology. All right, so that wraps up renal histology. If you have any questions, concerns, feedback, let us know in the comments, or you can message any of us directly. Otherwise, we'll see you in the next one.